Hi everyone, Kevin here with lecture 27. We're going to talk about ATP synthase, and then we will talk a little bit about how food other than glucose enters central metabolism. We won't cover fermentation in this set of videos because that was covered in a separate lecture. So we'll look first in this video at ATP synthase. Then in video number 2, we will look at how food other than glucose, that is, other catabolic pathways, how those link into central metabolism. And then we will not do topic 3 because we've looked at that in other videos. So, how does ATP synthase work? We are into the end of central metabolism now. So, we looked at the electron transport chain a little bit when we talked about reduction potentials, and we looked at all the different reduction electron centers within the electron transport chain and the reduction potentials of those electron centers. And we talked about how as electrons flow, protons can be pumped from the matrix side of the membrane in the mitochondria, the inner membrane, to the intermembrane space between the inner and outer membranes of the mitochondrion. So electrons come in, they come in at complex 1 or complex 2. Protons, they flow down the chain, and protons are pumped as the electrons flow. All right, so we know that pretty well. When the proton gradient is built up, and that's shown by all of these plus charges indicating the protons are in higher concentration in the intermembrane space. When the proton gradient builds up, those protons flow back through ATP synthase, shown here on the lower right in this image. So ATP synthase then produces ATP, and here's the overall reaction: ADP plus an inorganic phosphate, termed here just PI. The phosphate is hooked onto adenosine diphosphate to make adenosine triphosphate. And so there's our production of ATP. So everything we've done, harvesting electrons from glucose through glycolysis, and then we went through the pyruvate dehydrogenase complex, and then through the TCA cycle. Throughout there, we, on a stepwise basis, oxidized glucose. And we took the electrons that resulted from that oxidation of glucose, we put those on an NADH or FADH2, and those were dumped on the electron transport chain. We pumped protons, and this is where it all culminates in the use of ATP synthase to actually make ATP out of that proton gradient, or from using the power of that proton gradient. So as protons flow just by diffusion back from the intermembrane space into the matrix, they flow through ATP synthase and ATP is produced. So how does that happen? How do protons turn ATP synthase? Well, in this picture, this is ATP synthase, and you notice it has several different parts. We got alpha and beta subunits in this structure at the top. We've got a little gamma subunit here, and then we have this large part that's embedded in the membrane down below. Protons actually bind in this part embedded in the membrane. That turns, and that turns this gamma rotor, which causes the upper part here to actually turn and change conformation. And as it turns and changes conformation, we know oftentimes change in conformation generates chemical reactions, or vice versa. Sometimes chemical reactions generate changes in conformation. In this case, the change in conformation catalyzes the reaction that takes inorganic phosphate and puts it on ADP to make ATP. So protons bind on the P side of the membrane. This is the inner I-N-N-E-R membrane of the mitochondrion. The P side is in the intermembrane space, and protons flow back through to the N side, which is in the matrix of the mitochondrion. The way it works is when protons bind, when they come into this C10 segment of ATP synthase, and the protons bind, they actually create steric interference. In other words, they fill up space, and they cause part of C10 to push off the A domain. So you see the A domain here in the picture. C10 actually pushes off the A domain in response to protons binding. So you imagine this tiny little proton, subatomic particle, the nucleus of a hydrogen atom. All it takes is for that to bind, for the C10 domain to push off the A domain. That proton creates steric interference. C10 turns. This turns the gamma domain, which changes the conformation of the alpha and beta domains up at the top, as I mentioned. The changes then catalyze the production of ATP, as I also mentioned. So here's a look, kind of a top-down look at the alpha and beta domains of ATP synthase. So we're looking down on that gamma domain in the middle, and it's connected to the alpha and beta domains. And there are 3 sets of alpha and 3 sets of beta domains here around the circle. And the alpha and beta domains, as they rotate around, they either contain ADP, or they're empty, or they contain ATP. So they start out empty. As the circle turns, the conformation changes. The first change in conformation catalyzes the pickup of ADP. As it turns further, ADP picks up a phosphate and turns into ATP. So the conformation continues to change. Sorry about these stray lines here in my inking of this slide. As it continues to turn, the conformation changes, and we go from empty to ADP to ATP. So the loose conformation binds ADP and the inorganic phosphate. The tight conformation binds ATP so tightly that it's formed from ADP and PI. In other words, it brings ADP and PI together and catalyzes the reaction of those two to form ATP. So this is where all of that energy that was stored from food, that is used to make a proton gradient, it is used to turn this around the circle, change the conformation, and produce ATP. So this is how we get from the chemical energy stored in food to the energy stored in the molecule ATP, which then we can use to do things in the body. The empty conformation then releases ATP, and then we go back around the circle. Here is an animation. Actually, it's not an animation. It's just an image that shows ATP synthase. It's pretty well done. If you want to check it out, I'll put the link in Canvas. Here again is another look at the turns, and this breaks it into the 3 parts. And so you can see the protons coming on the P side and leaving on the N side. So protons come on the P side, they leave on the N side, and this is showing you see the direction of the arrows. You can see the way things are turning. But look at this, look at the steps, and trace each of the subunits as they go around the circle and see how they change to get a feel for that empty, the binding, and the creation and release of ATP. We were able to actually see this happen, and Paul Boyer, whose picture is on the front page of these slides, he died this year at age 99. He got the Nobel Prize along with one other researcher for discovering ATP synthase and how it works. And one of the things researchers did is they actually attached to this whole little molecular motor a an actin filament on the end that you could actually visualize when you saw the ATP synthase motor turning. Here then is a look at ATP synthase turning in the electron microscope. So you can see how that actin filament, which is showing up in this micrograph, you can see it turning. So it's gone from sort of horizontal here in the middle of the top row, horizontal on the upper left. It starts to turn as you go from left to right. So go from top left to bottom right, and you can see that little actin filament turning around, and that is the turning of ATP synthase. A pretty cool look at an actual enzyme. It's big enough to see in this with this technique. Here's a link to a YouTube video that is a really nice look at ATP synthase and showing it turning, and it even indicates the change in conformation. So please watch this. The link is in the module 27 on Canvas. That's it. Thanks for watching video number 1. There's one more video to finish off lecture 27. See you then.